Hello everyone, welcome to Tawheed Foundation for Allied Healthcare Sciences Bangalore. Today's class is about Immunology Part 6. Topic is Immune System for BSCMLT third year student. Immune system. So today I'll be discussing an important topic that is cell development in the immune system, focusing on B lymphocyte, T lymphocyte and natural killer cells. Before we get into the details, let's understand these terms. Our immune system is like the body's personal defense army. It protects us from bacteria, viruses, and other foreign invaders. To do this effectively, it relies on specialized cells that are trained to recognize, attack, and remember these threats. Among these immune cells, B cells, T cells, and NK cells play the most crucial roles. Each of them develops in a specific organ, has a unique function, and contributes differently to keeping us healthy. So, in today's class, we'll look at how these cells develop, where they mature, and how each one helps in building our body's defense mechanism. So let's begin with introduction to the lymphoid system. The lymphoid system is the structural and cellular framework of the immune system. Immune cells are not scattered randomly. They are organized into specialized tissues and organs that allow efficient immune surveillance, communication, and response. These collectively form the lymphoid system. Its major cellular components are lymphocytes, macrophages, that is antigen presenting cells, and epithelial cells that provide structural support or regulatory signals. Depending on their arrangement, these immune cells form either capsulated lymphoid organs or like thymus, spleen, and lymph nodes, or diffuse accumulations such as the mucosa associated lymphoid tissue, that is MALT, present in the lining of the gut, respiratory, and genitourinary tracts. This organization ensures that the body can detect and respond rapidly to pathogens entering through different routes. So, these immune cells are organized into tissues and organs for efficient function. Together, these will form lymphoid system. Components include lymphocyte, macrophages, and epithelial cells. These cells are arranged as capsulated organs or diffuse accumulation. Capsulated example is thymus, spleen, and lymph nodes. Diffuse accumulation example is MALT in mucosal tissues. Next is classification of lymphoid organs. Lymphoid organs are broadly divided into primary, that is central lymphoid organs, and secondary, that is peripheral lymphoid organs, the thymus and bone marrow. So, primary and secondary lymphoid organs types based on their functions in immune cell development and activity. Primary lymphoid organs, the thymus and bone marrow or fetal liver in early life, are the sites where lymphocytes originates, mature, and become immunocompetent. T cell mature in the thymus, while B cells mature in the bone marrow. Secondary lymphoid organs, including the spleen, lymph nodes, and mucosa-associated lymphoid tissue, that is MALT, are where mature lymphocytes encounter antigens, become activated, and initiate immune responses. This division reflects the two-step nature of immunity. First, the education of immune cell, and second, their deployment for defense. So, Lymphoid organs are classified into two main types. First is primary and second is secondary. Primary is central lymphoid organs, whereas secondary is peripheral lymphoid organs. Peripheral lymphoid organs. So in primary, it is a site of lymphocyte formation, maturation, and differentiation. It includes thymus and bone marrow or fetal liver. In secondary, it is a site of immune response activation. It includes spleen, lymph nodes, and MALT. So we'll see primary lymphoid organ. Primary lymphoid organs are the training grounds for lymphocytes. Here, lymphoid stems cells proliferate and differentiate into functional T or B cells. During this process, they rearrange their antigen receptor genes, allowing them to recognize specific antigens later in life. In mammals, T cell mature in thymus while B cell mature in the bone marrow. In bird, the bursa of Fabricius serves as the B cell maturation site, hence the name B cell. Once fully mature, lymphocytes leave these organs occupied with antigen receptors but still naive, meaning they have not yet encountered their specific antigen. This stage ensures the immune system has a diverse repertoire of cells ready to respond to future infection. So, what is primary lymphoid organs? Here it is a major site of lymphopoiesis. Here lymphocytes differentiate from stem cells and mature into functional immune cell. T cells mature in the thymus, whereas B cell mature in bone marrow or bursa of Fabricius birds. That is why it names B cells. Lymphocytes here acquire antigen receptors, preparing them for future antigenic challenge. Thymus structure. The thymus is a below-bed organ located behind the sternum in the anterior mediastinum. Each lobe contains numerous lobules separated by connective tissue trabeculae. Historically, each lobule shows two distinct zones, that is, cortex densely packed with immature T cells called thymocytes, medulla containing more mature T cells and supporting epithelial cell at the corticomedullary junction. There are numerous interdigitating dendritic cells, that is IDCs, and macrophages which play vital roles in T cell education. These structural compartmentalization supports sequential stages of T cell development and selection, filtering out self-reactive or defective cells before they enter circulation. So thymus, it is a below-bed organ located in the upper chest, that is anterior mediastinum. Each lobe has multiple lobules separated by connective tissue, trabecule, cortex, and medulla. Cortex is outer region which is densely packed with immature thymocytes, and medulla contain mature T cells and epithelial cell. Corticomedullary junction, it is rich in interdigitating dendritic cell, that is IDCs, and macrophages. Next is thymus function and cell type. The thymus functions as the central organ for T cell maturation and selection. The epithelial cells form an intricate network that secretes several thymic hormones and cytokines such as thymulin, thymopoietin, and thymosin. So thymulin, thymopoietin, and thymosin, and thymus expressed chemokine, that is TECK, that attracts and guides precursor T cells to mature. Within thymus, T cell undergo two critical selection processes. First is positive selection, ensuring T cell can recognize self-MHC molecules, and negative selection, eliminating those that react strongly to self-antigens. The interdigitating dendritic cell, that is IDCs, and macrophages assist by presenting self-MHC molecule during this processes. The result is a pool of mature CD4 helper, CD4 plus helper and CD8 plus cytotoxic T cells that can recognize foreign antigens in the context of self-MHC, a cornerstone of adaptive immunity. So, epithelial cells form a network secreting factors that attract and mature T cells. Secreted factors include thymus expressed cytokine, that is TECK, thymulin, thymopoietin, thymic humoral factors, and thymosin. So these thymic hormones were secreted. IDCs and macrophages from bone marrow present MHC molecules for T cell selection and T cell maturation, first immature and then it mature CD4 plus, that is helper, and CD8 plus, that is cytotoxic T cells capable of antigen recognition. Next is thymic involution. The thymus is highly active during infancy and childhood, reaching maximum size and activity around puberty. So this thymus is most active during childhood and puberty phase. After that, it undergoes age-related involution, where much of the thymic cortex is replaced by adipose and connective tissues. However, some thymic tissue remains functional throughout life. So, after puberty, it undergoes age-related involution and cortex atrophies, medulla persists with residual function. So, even in adults, a reduced but continuous output of new T cells occurs, maintaining immune surveillance and memory. The process of involution does not signify total loss of function, but rather a transition to a maintenance phase of immune competence. Clinically, this explains why older adults may have slower immune responses and a reduced ability to recover from new infections. So, T cell generation continues in adult but at a reduced rate. Despite involution, thymus remains vital for maintaining immune competence. Next is bone marrow and fetal liver B cell development. In mammals, bone marrow serves as the major site for B cell development, while in embryos, the fetal liver performs this function. In birds, the bursa of Fabricius is the equivalent organ, hence the term B cell. Bone marrow provides both the lymphoid stem cell that give rise to B and T lineages and the microenvironment needed for B cell maturation, including stromal cells and cytokines such as IL-7. During development, B cells undergo immunoglobulin gene rearrangements to produce unique antigen receptors. Those that successfully form functional receptors mature and migrate to peripheral organs. The bone marrow thus ensures that only properly formed and non-self-reactive B cells enter the circulation. So in birds, B cell will develop in bursa of Fabricus, hence it is named B cells. In mammals, fetal liver, that is during embryonic life, and bone marrow, which is after birth, are sites for B cell differentiation. Bone marrow will provide lymphoid stem cells for both B and T lineages, microenvironment for B cell maturation, and antibody gene rearrangement. Next is secondary lymphoid organ. Secondary lymphoid organs are the site of immune activation. Once lymphocytes mature in primary organs, they migrate via blood and lymph to these peripheral sites where they can encounter antigens. Key organs include the spleen, lymph nodes, and mold, which encompasses tonsils and Pierce patches. These tissues provide an ideal microenvironment for interaction between antigens, anti antigen presenting cells and lymphocytes activation. They act as meeting grounds for immune recognition, trapping antigens, promoting clonal expansion of specific lymphocytes, and generating effector cells and antibodies. So, secondary lymphoid organs will include spleen, lymph nodes, and mild. Example is tonsils and Pierce patches. Its function is to initiate immune responses upon antigen encounter. It contains lymphocytes, macrophages, dendritic cells that interact with antigen. It serves as sites of antigen trapping, lymphocyte activation, and clonal expansion. Different secondary organs respond to different routes of antigen entry. Secondary organs functional roles. So each secondary lymphoid organ has a specialized role depending on how antigens enter the body. The spleen filters blood-borne antigens, making it essential against systemic infections. Lymph nodes filter lymphatic fluid from tissues, capturing antigens that enter through the skin or mucosal surfaces. Mild, including GALT, that is gut, and BALT, that is bronchial, monitors mucosal entry points such as the respiratory, digestive, and urogenital tracts. Here, the key defense molecule is secretory IgA, that is SIGA, which is secretory IgA, which neutralizes pathogens directly at mucosal surfaces. Next is this SIA is the key effector molecule in mucosal immunity. This compartmentalization ensures that the immune system can defend every possible route of entry efficiently. Next is lymph node structure. Lymph nodes are small bean-shaped structures ranging from 2 to 10 mm in size. They are encapsulated by fibrous connective tissues that extends inward as trabecular lymph enters via afferent lymphatic vessels and exists through efferent lymphatics at the hilus where blood vessels also pass. Internally, the node is divided into cortex, paracortex, and medulla. Cortex, mainly B cell follicles, primary and secondary. Paracortex, T cell rich zone containing antigen presenting cells, APCs, dendritic cells with MHC2. Medulla contains plasma cell, macrophages, and some lymphocytes. This organized structure ensures that lymph-borne antigens meet the right immune cells for activation. So lymph node, it is small round or kidney-shaped organs ranging in size 2 to 10 mm. Hilus, it is nothing but it will help in indentation where blood vessels and afferent lymphatics exist. Afferent lymphatics bring lymph into the node. It is surrounded by fibrous capsule with trabeculae extending inward. Internal region contains these three parts: cortex, paracortex, and medulla. Cortex, B cell area, forms follicles. Paracortex, T cell area rich in APCs and MHC2. Medulla, plasma cell, macrophages, and some lymphocytes. Next is lymph node function. So lymph node act as the biological filters for lymphatic fluid, trapping microorganisms, foreign particles, and tumor cells. Within the cortex, primary follicles contain resting B cells, while secondary follicles form germinal centers following antigen exposure, sites of B cell proliferation, differentiation, and antibody production. Macrophages in the node phagocytose debris and pathogens, while dendritic cells present processed antigen to T cells. The nodes also facilitate lymphocyte circulation and proliferation. During active immune responses, lymph nodes enlarge, a condition known as lymphadenopathy due to proliferation of activated lymphocytes and plasma cells. So, function of lymph node. First primary follicle, it is a resting B cell phase. Secondary follicle, it is an active B cell forming a germinal center after antigenic stimulation. Its function is filter lymph for foreign materials and microorganisms, phagocytosis by macrophages, and lymphocyte proliferation and circulation. This lymphadenopathy is nothing but lymph nodes enlarged during immune response. Next is spleen structure. The spleen is the largest secondary lymphoid organ performing functions similar to a giant lymph node but for blood rather than lymph. It is enclosed by a delicate fibrous capsule with trabeculae extending into the anterior. The splenic artery branches into trabecular arteries, which further divide into central arterioles, surrounded by lymphoid tissues forming the white pulp. The white pulp contains PALS, that is periarteriolar lymphoid sheath, a T cell zone, and B cell follicles with adjacent marginal zones rich in macrophages. Surrounding this is the red pulp, a network of blood-filled sinusoids lined by macrophages and reticular cells responsible for filtering and recycling blood cells. So spleen, it is the largest secondary lymphoid organ. It filters blood instead of lymph. It is covered by fibrous capsule with trabeculae extending inward. Splenic artery branches into trabecular arteries, then central arterioles. White pulp, it is a lymphoid tissue around arterioles containing pulse and follicles and marginal zones. Red pulp, it is a vascular sinusoids with macrophages and reticular cells, filters RBCs and pathogens. Spleen function. The spleen's dual roles are hematological and immunological. It filters the blood to remove aged, damaged, or infected red blood cells and platelets. The red pulp serves as a storage and filtration site, recycling iron and hemoglobin components. Simultaneously, the white pulp is a site of immune surveillance where antigens from the blood encounter lymphocyte and macrophages, initiating immune responses. Almost half of the body's blood volume passes through the spleen daily, allowing constant monitoring for blood-borne pathogens. Thus, splenic dysfunction or removal increases susceptibility to bacterial sepsis. So, spleen's function is to filters blood, immune surveillance, produces immune responses, recycle iron and hemoglobin, stores blood cells, and half of total body volume. So, it filters blood, removes aged, damaged, or infected blood cells. In immune surveillance, lymphocytes, macrophages, and dendritic cells detects pathogens. It produces immune responses against blood-borne antigens. It recycles ions and hemoglobin from old red cells. It stores blood cells and platelets as a reserve supply. It is nearly half of total body volume passes through the spleen daily for immune inspection. Next is mucosa associated lymphoid tissue, that is MALT. MALT comprises scattered but functionally integrated non-encapsulated lymphoid tissues in mucosal lining, sites most frequently exposed to pathogens. Subtypes include GALT, that is gut associated, Peyer's patches, appendix, intestinal lymphoid aggregates, BALT, that is bronchus associated, respiratory tract mucosa, genitourinary mucosa, that is genitourinary malt, urogenital linings. Malt contains B anti-lymphocytes, plasma cells, and macrophages that generate secretory IgA, SIgA, providing the first line of defense by neutralizing pathogens before they penetrate deeper tissues. This distributed system ensures localized immune protection without causing widespread inflammation. So MALT is non-encapsulated lymphoid accumulation. It is located in mucosal linings, that is GALT, BALT, and genitourinary mucosa. GALT, it is gut associated. It includes intestines and Pierce patches. BALT, it is bronchus associated, that is respiratory tract. It contains B cells, T cells, plasma cells, and macrophages. It produces secretory IgA for local mucosal defense, and it protects body from pathogens entering through mucosal surfaces. So this lymphoid organs at a glance. Lymphoid organs. So here, to summarize, the lymphoid system functions as a coordinated network ensuring continuous immune surveillance and defense. First is primary lymphoid organs. The thymus and bone marrow are the developmental sites where immature lymphocytes become immunocompetent. Second is secondary lymphoid organs, that is lymph nodes, spleen, and MALT are where these mature lymphocytes encounter antigens, activate, proliferate, and mount immune responses. So, together, these organs maintain immune readiness. The primary organs generate soldiers, while the secondary organs are the battlefields where the immune wars are fought and won. Key points: lymphocytes mature in primary organs, it becomes functional. Immune responses occur in secondary organs after antigen exposure. Together, these ensure constant immune surveillance and defense. Next is subepithelial lymphoid organs. Beneath the mucosal surface of our body, especially in the alimentary canal, respiratory tract, and genitourinary system, these are dense collection of immune cells known as subepithelial lymphoid organs. These include T lymphocyte and B lymphocytes, plasma cells, and macrophages. Their main function is to detect, trap, and respond to any foreign substances that enter through these mucosal surfaces, which are the most common entry points for pathogens. These lymphoid tissues can appear in two forms: diffuse unorganized aggregates that are scattered beneath mucosal linings, or discrete nodular structures such as the tonsils in the pharynx and Peyer's patches in the intestine. Together, they act as a sentinels that provide an early immune defense before systemic responses are triggered. Next is tonsil structure. The tonsils are masses of lymphoid tissue situated beneath the stratified squamous epithelium of the nasopharynx and soft palate. Unlike other lymphoid organs, they are encapsulated, meaning there is no fibrous capsule separating them from the surrounding tissues. This unique structure allows antigens from food and air to come in direct contact with immune cells. Tonsils are rich in macrophages, B cells, T cells, and plasma cells, which together coordinate immune responses. Anatomically, the tonsils form part of Weldeyer's ring, a circular arrangement of lymphoid tissue consisting of the pharyngeal, palatine, and lingual tonsils. This ring forms a protective barrier at the gateway to the respiratory and digestive tracts, making it one of the body's first line of defense against airborne and ingested microbes. So tonsils, it is aggregates of lymphoid tissues located beneath stratified squamous epithelium of the nasopharynx and soft palate. It is encapsulated, that it means no fibrous capsule is present. It is rich in macrophages, B cell, T cell, and plasma cell. It forms part of Weldeyer's ring, that is pharyngeal, palatine, and lingual tonsils. Function of tonsils. Functionally, the tonsil act as the immune surveillance organs that detect pathogens entering through the mouth or nasal cavity when foreign particles or microorganisms are detected. Antigen presenting cells activate B lymphocyte, which differentiate into plasma cells that secrete secretory immunoglobulin A, that is SIgA. This antibody coats the mucosal surface and neutralizes microbes before they can invade deeper tissues. Thus, tonsils not only provide the local protection but also serve as the first site of adaptive immune activation, helping to generate immunological memory. Because of their location, they are often the first lymphoid organs to encounter environmental antigens from inhaled or ingested materials. Next is Peyer's patches structure. Peyer's patches are specialized aggregates of lymphoid tissue found in the submucosa of the ileum, part of the small intestine. Structurally, they contain multiple lymphoid nodules, each composed of B cell rich follicles surrounded by T cells and macrophages. Overlying the Peyer's patches are specialized epithelial cells known as M cells or microfold cells, which function to transport antigens from the intestinal lumen to the underlying immune cells. Like tonsils, Peyer's patches lack a capsule, allowing close interaction between the mucosal surface and the immune system. This structural arrangement ensures constant monitoring of intestinal contents for foreign antigens. So Peyer's patches, it is found in submucosa of the ileum, which is small intestine. It is consists of aggregated lymphoid nodules, that is B cell, rich follicles plus T cells plus macrophages. It is covered by specialized M cell, that is macrophage cells for antigen support. It lacks a capsule. It is similar to tonsils. Peyer's patches functions. The Peyer's patches are critical components of the gut-associated lymphoid tissue, that is GALT. Their primary function is to detect antigens that pass through the intestinal epithelium and to initiate mucosal immune responses. Upon antigen recognition, they stimulate B cells to produce secretory IgA, that is SIgA, antibodies that are secreted into the gut lumen. These antibodies neutralize pathogens and toxins, preventing their attachment and invasion. Additionally, Peyer's patches generate memory B cells, ensuring long-term protection against intestinal pathogens. They help maintain gut immune homeostasis, providing a balance between immune defense and tolerance towards beneficial gut microbiota. So Peyer's patches function is to detect antigen that diffuse across the intestinal epithelium, to induce production of mucosal antibodies, to generate local memory cells for mucosal protection, to critical in maintaining gut immune homeostasis, that is part of GALT. Mild mucosa associated lymphoid tissue. The mucosa-associated lymphoid tissue refers to all organized and diffuse lymphoid tissues found beneath mucosal linings throughout the body. Mild includes specialized forms such as GALT, gut-associated, and BALT, bronchus-associated lymphoid tissues. The key function of mild is to produce SIgA antibodies, which act as a chemical shield across mucosal surfaces, neutralizing bacteria and viruses before they penetrate epithelial barriers. Malts thus serve as a first-line immune barrier, providing local immunity at all potential mucosal entry sites such as the gastrointestinal, respiratory, and urogenital tracts, protecting against environmental antigens and pathogens. Next is skin and immune surveillance. The skin, though primarily a physical barrier, also performs important immunological functions. Distributed within the epidermis and dermis are populations of T lymphocyte, macrophages, and specialized antigen presenting cell known as Langerhans cell. This is specialized antigen presenting cells. These cells continuously monitor the skin for invading pathogens or tissue injury. Unlike lymphoid organs, the skin does not form distinct nodules. Instead, these immune cells remain scattered but active, providing continuous surveillance. This system is known as skin associated lymphoid tissue SALT. SALT plays an essential role in early pathogen recognition, antigen presentation, and activation of local immune responses, making the skin an integral part of both innate and adaptive immunity. Next is cellular composition. The lymphoreticular system consists of both functional immune cells and supporting structural cells. The main immune cells include lymphocytes, plasma cells, and a range of phagocytic cells such as macrophages, neutrophils, eosinophils, and dendritic cells. In addition, there are reticular cells, endothelial cells, and fibroblasts, which form the connective tissue framework and produce signaling molecules that regulate immune activity. Together, these cells maintain the architecture and functional integrity of lymphoid organs and coordinate immune responses throughout the body. Next is lymphocytes' general characteristics. Lymphocytes are small, round cells typically measuring 5 to 12 μm, that is micrometers in diameter. They possess a large, darky staining nucleus and a thin rim of cytoplasm. Although they appear similar under a microscope, lymphocytes and functionally diverse. They are classified into three major types, that is B cells, T cells, and NK cells based on origin and surface markers. T lymphocyte, which mediates cellular immunity, B lymphocyte responsible for antibody production, and NK, that is natural killer cells, which destroy infected or tumor cells without prior sensitization. These cells continuously circulate between the blood, lymphoid organs, and tissues, ensuring rapid detection of foreign antigens. Next is lymphocyte distribution. The distribution of lymphocyte types varies across different tissues. Approximately 75% of circulating lymphocyte are T cells, 15% are B cells, and 10% are NK cells in lymphoid organs such as the lymph nodes, spleen, and malt. Both T and B cells coexist to coordinate immune responses. In contrast, tissues like blood and thymus are predominantly populated by T cells, reflecting their role in immune regulation and activation. This strategic distribution ensures that immune defense mechanisms are present wherever foreign antigens may appear. Next is origin and development of lymphocyte. All lymphocytes originate from hematopoietic stem cells located in the bone marrow or during fetal life in the fetal liver. These stems commit to the lymphoid lineage, becoming lymphoid progenitor cells. Their growth and maturation depends on cytokines such as interleukin 7, IL-7, and stem cell factor, that is SCF. From this point, the development diverges. T cell precursor migrate to the thymus for maturation into functional T lymphocytes, while B cell precursor remain in the bone marrow to mature and express surface immunoglobulins. This differentiation marks the origin of the two adaptive immune arms, cell-mediated and humoral immunity. Next is circulation and function of mature lymphocytes. Once fully mature, T and B lymphocytes enters the bloodstream as naive lymphocytes, meaning they have not yet encountered their specific antigen. They circulate continuously between blood, lymph, and secondary lymphoid organs such as the spleen, lymph nodes, and tonsils. Within these organs, antigens are trapped and presented by antigen-presenting cells, allowing lymphocytes to recognize them. Upon recognition, the lymphocytes become activated, proliferate, and initiate an immune response. The organization of secondary lymphoid organs ensures that antigen lymphocyte interaction occurs efficiently and effectively. Activation and differentiation. Naive lymphocytes have a limited lifespan unless they are activated by a specific antigen. When activation occurs, the lymphocytes undergo clonal proliferation, producing numerous identical cells with the same antigen specificity. These cells then differentiate into two major types, that is effector cells and memory cells. Effector cells which perform immediate immune defense functions such as antibody secretion or cytotoxic killing. Memory cells which persist long-term and enable rapid secondary responses upon re-exposure to the same antigen. This mechanism of activation and memory formation forms the basis of adaptive immunity and long-lasting protection after infection or vaccination. Next is B lymphocytes. B lymphocyte B cell development. Here it begins in the fetal liver during embryogenesis and continues in the bone marrow after birth. The hallmark of B cell is their ability to synthesize immunoglobulins which function as antibodies. Each B cell expresses a unique B cell receptor BCR that recognizes a specific antigen. When an antigen binds to the BCR, that cell is activated, undergoes clonal selection and expansion, and differentiate into two types of progeny, that is plasma cells which produce and secrete antibodies, and memory B cells which provide long-term immunity. This entire process constitutes the foundation of humoral immune responses. Surface markers during B cell maturation. The development of B cells can be traced through the appearance of specific surface markers. Pro B cell express CD4 5R, CD19, CD43, CD24, and c-KIT, but lack surface immunoglobulins. Pre-B cell expresses CD25 pre-BCR receptor, indicating rearrangement of immunoglobulin genes. Immature B cell begins to express surface IgM, enabling antigen recognition. Mature B cell displays both IgM and IgD, making them fully functional and capable of circulating to secondary lymphoid organs. These markers changes are crucial for identifying each developmental stage and diagnosing B cell related diseases. Next is B cell subtypes. There are two main functional subtypes of B cell, that is B1 cell and B2 cell. B1 cell develop early in embryonic life and are primarily found in the pleural and peritoneal cavities. They express IgM and CD5, respond poorly to protein antigens but strongly to carbohydrate antigens, and produce low affinity natural antibodies that provide quick non-specific defense. B2 cells, on the other hand, are conventional B cells that make up about 95% of total B cells. These are generated continuously from the bone marrow after birth and are distributed throughout lymphoid organs. They produce high affinity specific antibodies during immune responses and form the mainstay of adaptive humoral immunity. Next is mild and lymphoreticular cells. So, in summary, the mild system, which includes tonsils, Peyer's patches, GALT and BALT, provide localized, localized immune protection at mucosal surfaces. The lymphocytes T, B, and NK cells act as the core component of adaptive and innate immunity. B cell generate antibodies and memory responses. T cells regulate and mediate cellular immune reactions, and macrophages and dendritic cells serves as antigen presenting cells that link, initiate, and adaptive immunity. Together, These elements form the lympho-reticular system, ensuring continuous immune surveillance and efficient protection against infections across all body surfaces and internal environments. Next is plasma cells. Plasma cells are the terminally differentiated effector cells of the B cell lineage, meaning they represent the final stage of B cell maturation. Unlike resting B cells, plasma cells do not express surface immunoglobulins. Instead, they are highly specialized for the production and secretion of large quantities of antibodies. These antibodies are released into extracellular environment where they neutralize pathogens, mark them for phagocytosis, or trigger complement activation. Plasma cells are thus critical for humoral immunity, serving as the main machinery for antibody-mediated defense. Although short-lived and non-dividing plasma cells represent the functional endpoint of B cell differentiation, providing immediate and antigen-specific protection. Next is structure of plasma cells. Plasma cells are typically oval or egg-shaped. Their nucleus is eccentrically placed and exhibit a characteristic cartwheel or clock-free chromatin pattern formed by dense clumps of chromatin around the nuclear membrane. This unique pattern is often used as histological hallmark for identification. The cytoplasm is abundant and basophilic, reflecting a high content of rough endoplasmic reticulum, that is RER, which is necessary for antibody synthesis. The Golgi apparatus is well developed and functions in packaging and secreting antibodies efficiently. Importantly, plasma cells lack surface immunoglobulins as the role is to secrete, not to bind antibodies. This structural specialization ensures that plasma cells are highly efficient antibody factories. Next is functions of plasma cells. The primary function of plasma cell is to secrete immunoglobulins that are specific to the stimulating antigens. These antibodies maintain their antigen binding capacity and mediate immune protection by neutralization, opsonization, and complement activation. Beyond antibody production, cells of the B lineage, including plasma cells, can also participate in antigen processing, presentation to T helper T cells, and cytokine secretion, which regulate the growth and differentiation of other immune cells. Plasma cells are terminally differentiated, meaning they do not divide further and their lifespan is generally short, but they provide a rapid and specific humoral response after B cell activation. Next is overview of T cells. T cells are lymphocytes derived from thymus and are primarily responsible for cell-mediated immunity, that is CMI. After maturation in the thymus, they become immunologically competent cells, ICCs, capable of recognizing antigens and mounting immune responses. Their major roles include antigen recognition, initiation of cell-mediated immune response, and retention of immunological memory. By circulating between blood and secondary lymphoid organs, T cells provide rapid and specific immune defense against intracellular pathogens such as viruses as well as tumor cells. Next is T cell receptors. T cells do not express immunoglobulins like B cells. Instead, they rely on T cells receptor TCRs to detect antigens. TCRs are membrane-bound heterodimers composed of two polypeptide chains. The majority of T cells express alpha and beta chains, alpha beta T cells, whereas a smaller subset expresses gamma and delta chains, that is gamma delta T cells. TCRs are never secreted. They are solely used for recognition of antigen fragment presented on MHC molecules of antigen presenting cells. This receptor is central to T cell activation and ensures antigen specificity and MHC restriction. Next is mechanism of antigen recognition by T cells. So T cells recognize antigens only when presented by antigen presenting cells, APCs, such as macrophages, dendritic cells, and B cells. Antigens are processed into peptide fragments which are loaded onto MHC molecules and displayed on the APC surface. The TCR then recognizes the peptide MHC complex, initiating T cell activation. This process is crucial for ensuring specificity and self MHC restriction, meaning T cell respond only to foreign antigens in the context of self MHC molecules. Next is types of immune responses mediated by T cells. So T cell mediated T cell mediated cell immunity T cell mediated immunity, that is CMI, by directly killing infected cells and activating other immune cells such as macrophages and cytotoxic cells. They also regulate immunity through helper functions. CD4 plus T cell stimulate B cells for antibody production and enhance cytotoxic T cell activity. By secreting cytokines, T cell modulate communication between immune cell and optimize the immune response, ensuring cooperation with macrophages for effective antigen presentation and pathogen destruction. Next is surface molecule on T lymphocytes. Mature T cells express a variety of surface molecules in addition to TCRs. These molecules are identified using monoclonal antibodies and are classified as cluster of differentiation CD markers. CD marker provide information about the stage of differentiation and functional specialization of T cells. Over 80 CD markers have been identified, but the most important for T cell functions include CD2, CD3, CD4, and CD8. These molecules are critical in signal transduction, antigen recognition, and adhesion. Next is key CD molecules on T cells. The key CD markers on T cells include CD2, present on all mature T cells, mediates adhesion and contributes to activation. CD3, associated with TCR, critical for signal transduction and initiating T cell activation. CD4, expressed on helper T cells, recognize antigens presented by MHC class II molecules. CD8, expressed on cytotoxic T cells, recognizes antigen presented by MHC class I molecule. These markers help define T cell subsets and determine their functional roles in immunity. Next is major T subsets. T cells are categorized based on surface CD markers. CD4 plus helper T cells constitute approximately 70% of circulating T cells and regulate immune responses. CD8 plus cytotoxic T cells comprise about 25% and mediate killing of infected or abnormal cells. Minor populations include double negative and double positive. Double negative CD minus 4 CD8 minus gamma T cells, that is 4%, involved in early mucosal defense, and double positive CD4 plus CD8 plus cells, that is 1% only, mainly observed during thymic development. Each subset has a distinct functional specialization and MHC restriction. Next is helper T cell, that is CD4 plus. CD4 plus helper T cells recognize antigens presented with MHC class II on APCs. Their main roles include activating B cells to produce antibodies, stimulating macrophages and cytotoxic T cells, and secreting cytokines or lymphokines that enhance the activity of other immune cells. They are often called master regulators or adaptive immunity due to their central role in coordinating both humoral and cell mediated responses. Next is cytotoxic T cell CD8 plus. CD8 plus cytotoxic T cell recognize antigen presented with MHC class 1 molecules on all nucleated cells. They are primarily responsible for killing virus, infected cells, tumor cells, and foreign particles, that is foreign graft cells. So, that cytotoxic mechanisms include the release of perforin, that is form spores, and gran enzyme that includes apoptosis. CD8 plus T cells are essential for cell mediated cytotoxicity and controlling intracellular pathogens. Next is minor T cell subsets. Double negative, that is CD4 minus and CD8 minus cells predominantly expresses gamma and delta TCR instead of alpha and beta TCR and provide early immune defense, especially in mucosal and epithelial tissues. Double positive CD plus, CD4 plus, CD8 plus cells are mostly present during thymic development and their function is peripheral tissues is not fully defined. These minor subsets highlight the diversity of T cell functions in both central and peripheral immunity. Next is overview of T cell maturation. T cell precursors originate from the yolk sac, fetal liver, or bone marrow and migrate to the thymus for maturation. The thymus provides a microenvironment with stromal cells and cytokines that guide the development, selection, and differentiation of T cells. During maturation, T cells acquire functional TCRs, express specific CD markers, and are selected for self-tolerance and MHC restriction, ensuring a repertoire capable of responding to foreign antigens while avoiding autoimmunity. Next is stages of T cell development. T cell development involves several sequential stages: pro T cell, pre T cell, immature T cell, and mature T cell. Pro T cell earliest stage expresses CD7. Pre T cell acquires CD2, synthesize CD3, begin TCR synthesis. Immature T cell expresses CD1, CD3, CD4, and CD8, develop either alpha beta or gamma delta T cells. Mature T cell loses CD1 and differentiate into CD4 plus helper or CD8 plus cytotoxic T cell ready to exit the thymus. These stages ensure proper development and functional competence. Next is selection process in thymus. The thymus perform positive and negative selection to generate functional self-tolerant T cells. Positive selection and negative selection. Positive selection ensure T cell can recognize self-MHC molecules. Negative selection eliminate T cells that react strongly to self-antigens, preventing autoimmunity. The result is a repertoire of T cells that are MHC restricted and ready to respond specifically to foreign antigens. Next is MHC restriction and functional differentiation. CD8 plus T cell recognize antigens in the context of MHC class 1, MHC class 1 leading to cytotoxic or suppressor function. CD4 plus T cells recognize antigens with MHC class 2 performing helper or inducer functions. Rare exceptions exist, but generally MHC restrict defines both recognition and effector function. Comparative features T cell, B cell, and macrophages. So this slide summarizes functional distinctions. T cells will express TCR, mature in thymus, mediate cell-mediate immunity, produce cytokines, and have CD2, CD3, CD4, or CD8 markers. B cell expresses surface IgBGR, mature in bone marrow, mediate antibody, mediated immunity, and have CD19, CD20 markers. Macrophages expresses MHC and FC receptors, mature form monocytes in bone marrow, perform antigen presentation and phagocytosis, and are activated to destroy pathogens. This comparison highlights the complementary roles of immune cells in innate and adaptive immunity. Next is null cells. So introduction to null cells. Null cells are a small subset of lymphocytes that lack classical T or B cell surface markers. They are also referred to as large granular lymphocyte, that is LGLS, and constitute about 5 to 10% of total circulating lymphocytes. Null cells include natural killer NK cells, ADCC cells, LK cells, and NK T cell. ADCC cells is antibody dependent cellular cytotoxicity. LK cells is lymphokine activated killer cells. NKT cell, that is natural killer cells. These cells are innate immune effectors acting as first line defenders against viral infections, intracellular bacteria, and tumor cells. Unlike T and B lymphocytes, null cells do not require prior antigen sensitization or MHC restrictions, allowing them to respond rapidly to abnormal or infected cells. Next is natural killer cells. NK cells are innate cytotoxic lymphocyte with the ability to kill target cells spontaneously without prior exposure or sensitization. They play a vital role in eliminating virus infected and tumor cells. NK cells are not MHC restricted and do not rely on antibodies for target recognition. Being the majority of null cells, they are rapid responders, forming a bridge between innate and adaptive immunity. NK cells identify target cells by detecting missing or altered self MHC class 1 molecule, a mechanism that ensures elimination of abnormal cells while sparing healthy tissues. So natural killer cell, it is innate cytotoxic lymphocyte with spontaneous killing ability. It kill virus infected and tumor cell without prior exposure, not MHC restricted and do not require antibodies. No antigen sensitization needed. It is a rapid responders. It will make up the majority of null cells. Surface markers of NK cells. NK cells are typically identified as CD16 plus, CD56 plus, or CD3 minus. CD16 receptor for Fc portion of IgG mediates ADCC. CD56 adhesion molecule used as a primary NK identification marker. CD3 and CD4 absent, distinguishing them from T cells. CD8 present on about 50% of NK cell but not essential for cytotoxicity. These markers profile differentiate NK cells from T lymphocyte, which are CD3 plus, CD16 minus, and CD56 minus. Next is mechanism of NK cytotoxicity. NK cell attach to glycoprotein receptors on target cell surfaces and release cytolytic factors. Perforin, it forms pores in the target cell membrane. Serine esterases TNF B lymphotoxin enters through these pores and induces apoptosis. Cytotoxicity is further enhanced by IFN, that is interferon. Functionally, perforin-mediated killing resembles the pore-forming activity of complement protein, that is C9, allowing NK cells to eliminate infected or malignant cells efficiently. Next is regulation of NK cells. NK cells activity is controlled by two receptor families, killer activation receptors and killer inhibition receptors. Killer activation receptors recognize carbohydrate or stress signals on target cells, triggering kill signals. Killer inhibition receptors bind to MHC1 molecule, delivering do not kill signals. So the balance between KAR and KIR determines whether the NK cell destroys or spares the target. Healthy cells with normal MHC1 are protected, whereas virus infected or tumor cells with reduced MHC1 are selectively eliminated. Next is NK T cell, that is a natural killer T cells. NK T cells are hybrid lymphocytes expressing features of both NK and T cells. They develop in the thymus and express a restricted alpha beta TCR. NK T cells recognize lipid or glycolipid antigens presented by non-classical MHC molecule, that is CD1D, and can rapidly secrete cytokines, especially IL-4 and IFN gamma. These cells are crucial for immune regulation, anti-tumor defense, and bridging innate and adaptive immunity. Next is ADCC cells, that is antibody dependent cellular cytotoxicity. ADCC cells are large granular lymphocyte expressing CD16 that bind to the FC portion of IgG antibodies attached to target cells. Once bound, they release perforins and granzymes, lysing antibody-coated cells such as bacteria, parasite, tumor cells. Unlike NK cells, ADCC cells rely on antibodies for guidance, forming a key component of antibody-mediated cytotoxicity. They are sometimes called killer K cells due to this cytotoxic function. Next is LK cell, that is lymphokine activated killer cells. LAK cells are NK cells activated by IL-2 produced by helper T cells. They exhibit enhanced cytotoxic activity against tumor cells and secrete cytokines such as IFN gamma, TNF alpha, GM-CSF, and CSF1. They express activation markers like NKp44 and are used clinically in adoptive immunotherapy for cancer such as the renal carcinoma. LK cells demonstrate how immune activation can be harnessed therapeutically. Now, introduction to phagocytosis. Phagocytosis is the oldest evolutionary defense mechanism, enabling ingestion and destruction of microbes and debris. In higher organisms, it is performed by specialized phagocytes rather than for nutrition. The process involves chemotaxis, attachment, ingestion, and digestion. Major phagocytic cells include macrophages, neutrophils, eosinophils, and dendritic cells, all contributing to innate immunity. Next is macrophages, origin and distribution. Macrophages originate from monocytes in the blood that migrate into tissues. They differentiate into tissue-specific macrophages, Kupper cell, liver, alveolar macrophages, lungs, Langerhans cells, that is skin, osteoclasts, that is bone. So macrophages are long-lived and can persist for months, serving as sentinels for pathogens detection and antigen presentation. Next is function of macrophages, that is phagocytosis. Macrophages are primary defenders attracted to infection sites via chemotaxis. They engulf pathogens to form phagosomes, which fuse with lysosomes to form phagolysosome for digestion. Phagocytosis is enhanced by opsonization, where IgG or C3b coat pathogen. Some microorganisms like Mycobacterium tuberculosis can resist digestion, highlighting the challenge of certain infections. Next is functions of macrophages to activation. Macrophages are activated by microbial products. The example is endotoxin, complement proteins, and interferons gamma from T cells. Activated macrophages exhibit enhanced metabolism, increased phagocytosis, and greater microbicidal and antigen presenting capacity, making them crucial for both innate defense and adaptive immunity. Next is functions of macrophages 3, that is antigen processing. Macrophages process antigens and display them with MHC molecules to helper T cells. This interaction is essential for T cell activation, bridging innate and adaptive immunity. By presenting antigen, macrophages initiate antigen-specific responses and support immune memory formation. Next is function of macrophages 4, that is cytokine production. Macrophages secrete biologically active molecules, including hydrolytic enzymes, binding proteins, and cytokines. Binding proteins like fibronectin and transferrin, and cytokines like TNF, IL-1, IL-6, and CSF. IL-1 acts as an endogenous pyrogen and stimulates IL-2 production by T cell Cytokine secretion contributes to inflammation, tissue repair, and regulation of immune responses. Next is neutrophils. Neutrophils are the most abundant white blood cells and key in acute inflammation. They are actively phagocytic but non-specific, forming the first line of defense in innate immunity. Their activity is enhanced by opsonins, that is IgG complement. Neutrophils are short-lived and their death contributes to pus formation at infection sites. Eosinophils are prominent in allergic reaction and parasitic infection, slightly larger than neutrophils, that is around 12 to 17 μm. Their granules contain eosinophil peroxidase, major basic protein, that is MBP, and charcot-laden crystal protein. They kill large parasites like schistosomes and trichinella phagocytose antigen antibody complexes and release cytotoxic granules extracellularly, contributing to antiparasitic and anti-allergic responses. Next is basophil and mast cell. Basophil, that is blood, and mast cell, that is tissue, contain histamine, heparin, and serotonin. Upon allergen exposure, they degranulate, releasing mediators that cause anaphylaxis, urticaria, and inflammation. These cells play a central role in type 1 hypersensitivity reactions, including asthma and allergic disorders. Next is dendritic cells. Dendritic cells are professional antigen presenting cell, that is APCs, found in almost tissues, especially at surface epithelia and lymphoid organs. They have dendritic projections and express MHC class II molecules. Although largely non-phagocytic, dendritic cell activate naive T cell and initiate primary adaptive immune responses, making them crucial for immunity. Next is Langerhans cells. Langerhans cells are epidermal dendritic cells constituting around 5% of all epidermal cells. They capture antigens entering through the skin, process them, and present them to T cell in regional lymph nodes, acting as immune sentinels. They provide early warning against pathogens and maintain cutaneous immune surveillance. Next is comparison of phagocytic cells. So cell type, this table highlights functional differences and specialization among phagocytic. So macrophages, its major function is phagocytosis, antigen presentation, and cytokine secretion. And its special feature is it is long-lived MHC2 molecule. Neutrophils, it is a rapid non-specific phagocytosis and it is first line of defense. Eosinophil, it will kill parasites and allergic response, and MBP and peroxidase granules. Basophil mast cell, its function is it has allergy and histamine will release, and special feature is degranulation. Dendritic cell major function is antigen presentation. Its special function is initiate adaptive responses. Next is null cells. So NK T cells, NK cells, ADCC cells, LAK cells, and NKT cells. So its function, target, and mechanism. NK cell, it is spontaneous target. It will target virus infected or tumor. Its mechanism is perforin plus apoptosis. ADCC cells, activation is IgG coated cells. Its target is antibody coated targets. Its mechanism is Fc receptor mediated killing. LAK cells, its activation is IL-2 activated NK cells. Its target is tumor cells. Its mechanism is cytokine release. NKT cells, its activation is lipid antigens. Its target is regulatory and tumor response, and mechanism is cytokine burst. Clinical correlation: NK cell deficiency, recurrent viral infection. Macrophages dysfunction is defective antigen presentation and immune compromise. Eosinophilia, it's parasitic infection or allergy. Excessive basophil and mass activation is asthma, urticaria, and anaphylaxis. Dendritic cell overactivation is autoimmune inflammation. This correlation demonstrates the clinical significance of innate immune cells and their role in disease pathogenesis. Next is MHC. So the major histocompatibility complex MHC is a group of genes that encode cell surface glycoproteins historically recognized as transplantation antigens. These molecules were initially identified because of their role in graft rejection, where tissues from genetically different individuals were rapidly attacked by the immune system. Later research revealed that MHC molecules plays a central role in immune recognition and antigen presentation, serving as a molecular interface for T cell mediated immunity. MHC genes are present in all vertebrates. In humans, the MHC is called human leukocyte antigen, that is HLA complex, whereas in mice it is referred to as the H2 complex. This system is fundamental in distinguishing self from non-self, forming the basis for immune response initiation. Next is historical significance. The MHC was first detected during tissue transplantation experiments, where it was observed that grafts between genetically non-identical individuals were rejected. The molecules responsible were termed transplantation antigens. The discovery of MHC revolutionized immunology, shifting it from a descriptive science to a molecular understanding of immune regulation. We now know that MHC molecules are critical for T cell antigen recognition and genetic variability at MHC loci. Explain why grafts are rejected in some individuals but tolerated in others. This historical perspective illustrates the connection between clinical observations and molecular immunology. Next is gene location and organization. MHC genes are located on the short arm of chromosome, that is 6p21.3 in humans. The region spans approximately 4 megabases and contains around 200 genes. These genes are organized into 4 main regions, that is A, B, C, and D, which are further divided functionally into 3 classes, that is first, second, and third. Each region encodes specific proteins with distinct immune functions, including antigen presentation, complement activation, and cytokine production. The MHC locus is one of the most gene-dense and polymorphic regions in the human genome, reflecting its critical role in immune defense. Next is overview of MHC classes. MHC molecules are categorized into 3 classes: class 1, class 2, and class 3. Class 1, HLA, A, B, and C present endogenous antigens to CD8 plus cytotoxic T cell and are expressed on all nucleated cells. Class 2, HLA, DP, DQ, DR present exogenous antigen to CD4 plus helper T cells and are expressed on B cells, macrophages, and dendritic cells. Class 3 encode soluble proteins such as complement component, TNFS, and heat shock protein, which support immune and inflammatory responses but are not membrane bound. Thus, class 1 and class 2 are central to antigen presentation, while class 3 provides immune system support. Next is MHC class 1 gene structure. Class 1 molecule are encoded by HLA, HLB, and HLC. Structurally, they consist of heavy alpha chains and B2 beta microglobulin. So alpha chain, it is around 43 kDa encoded in the cell membrane, and microglobulin is around 11 kDa non-covalently associated and coded elsewhere. The alpha chain has 3 domains, that is alpha 1, alpha 2, and alpha 3 within alpha 1 and alpha 2, forming the peptide binding groove. Class 1 molecules are found on nearly all nucleated cell except villus trophoblast, enabling surveillance for endogenous peptides. including viral or tumor-derived protein. This allows CD8 plus cytotoxic T cells to detect and eliminate infected or malignant cells. Next is expression and function of MHC class 1. So MHC class 1 molecule are expressed on all nucleated cells, enabling constant immune surveillance. They present endogenous peptides, including viral and tumor-derived proteins, to CD8 plus cytotoxic T cells, initiating cell-mediated killing if expression is absent or reduced. Cell become target for NK cell-mediated, that is missing self-recognition. Therefore, MHC1 molecule balance immune activation and tolerance. Next is MHC class 2 gene and structure. MHC class 2 molecules are encoded by the HLA-D region, that is DP-DQ DR loci, and consist of two polypeptide chain, that is alpha around 34 kDa and beta, that is around 29 kDa. Each chain has two domains, alpha 1, alpha 2 and beta 1 and beta 2, forming a peptide binding cleft. Class 2 molecules are primarily expressed on antigen presenting cells, B cells, macrophages, and dendritic cells. They can also be included on endothelial cells by IFN, that is interferon delta gamma. Class 2 molecule present exogenous antigens to CD4 plus helper T cell, which is critical for adaptive immune activation. Next is MHC class 3 region, located between class 1 and class 2 regions. Class 3 generates do not encode membrane-bound molecules. Instead, they produce soluble immune proteins such as complement components C2, C4, and factor B, tumor necrosis factors, that is TNF alpha, TNF beta, heat shock protein, that is HSP70 family. These proteins support immune signaling, inflammation, and complement-mediated defense, enhancing overall immune responses. HLA polymorphism. HLA loci are highly polymorphic with multiple alleles per locus. Around 24 alleles at HLA-A, 50 alleles at HLA-B. Each allele encode a unique MHC molecules, contributing to individual variability in immune responses. This diversity enhances population-level pathogen recognition but also complicates organ transplantation due to potential graft rejection. Next is immune response, IR genes. It is located mainly in the HLA-DR locus of class 2. IR genes control immune responsiveness to specific antigen. They influence T cell activation and antibody production. Certain HLA-DR antigens such as DR1 and DR3 are associated with strong graft rejection reaction and autoimmune predispositions. Understanding these genes is crucial for transplant compatibility and disease association studies. Next is other MHC linked genes. The MHC region also contains non-immunological genes, including heat shock protein, that is heat shock protein 2, HSP2, 21 beta hydroxylase, that is steroidogenesis enzyme. Although their immune functions are less clear, these genes highlight the MHC as a genetic hub with potential cross-regulatory effects on immunity and metabolism. Next is functional significance summary. So class 1 present endogenous antigens to CD8 plus T cell cytotoxicity. Class 2 present exogenous antigens to CD4 plus T cells helper responses. And class 3 produces soluble immune mediators, that is complement and cytokines. Together, these classes ensure immune recognition, communication, and regulation, coordinating adaptive immune response effectively. Next is clinical relevance. Transplantation MHC mismatch leads to graft rejection. Autoimmune diseases, specific HLA types increases susceptibility. Example, HLA B27, that is ankylosing spondylitis, HLA DR3, that is SLE and Graves disease. Infectious diseases, MHC alleles influence pathogen susceptibility and vaccine response. Highlight that HLA typing is critical in transplantation, autoimmune risk assessment, and predicting immune responses. So, summary. So, the lymphoid system is a network of organs, tissues, and cells that work together to defend the body from infection and foreign substances. It includes both central and peripheral structures where immune cells develop, mature, and function. These structures ensure the body can recognize, respond to, and remember antigens effectively. Next is primary lymphoid organs. The primary lymphoid organs are the thymus and bone marrow. These are the main sites where lymphocytes are formed and mature into functional immune cells. T cells develop in the thymus while B cells develop in the bone marrow. Hence, the immune cells acquire the ability to recognize specific antigens through receptor formation. Third is secondary lymphoid organs. The secondary lymphoid organs include the spleen, lymph nodes, and MALT, which stands for mucosa associated lymphoid tissue. These are the sites where immune responses actually take place. They trap antigens, activate lymphocytes, and initiate defense mechanisms like antibody production or cytotoxic activity. Fourth is thymus. Thymus is a below bed organ located in the upper chest. It serves as the maturation site for T cells where they learn to distinguish self from non-self. After puberty, the thymus gradually shrinks, a process known as thymic involution, but still retains some immune activity throughout life. Next is bone marrow or fetal liver. The bone marrow in adults and fetal liver in embryos are vital for the development of B lymphocytes. Here, B cells undergo gene rearrangement that allow them to produce antibodies specific to various antigens. These organs also supply lymphoid stem cells for both B and T cell lineages. Next is spleen. The spleen is the largest secondary lymphoid organ and plays a key role in filtering the blood. It removes old and damaged red blood cells and also detects and responds to blood-borne antigens. By doing so, it helps produce immune responses against pathogens circulating in the bloodstream. Next is lymph nodes. Lymph node act as a filter for lymph, the fluid that carries immune cells throughout the body. They trap antigens and are major sites for lymphocyte activation, proliferation, and differentiation. During infection, they enlarge due to active immune cell division, a condition called lymphadenopathy. Next is MALT, mucosa-associated lymphoid tissue. It includes structures like tonsils and Peyer's patches. It protects mucosal surfaces of the respiratory, digestive, and genitourinary tract. MAL produces secretory IgA antibodies, which prevent microbes from attaching and invading throughout mucosal lining. Next is lymphocytes, core of immune defense. The main immune cells are lymphocytes, which include T cells, B cell, and natural killer cells. They form the core of the immune system's defense strategy, recognizing antigen-producing antibodies and killing infected cells. Together, they maintain immune surveillance throughout the body. Next is B and T cells. B cells transform into plasma cell, which secrete antibodies that neutralize pathogen. This is called humoral immunity. On the other hand, T cells T cells are of two main types, CD4 plus helper T cells, which regulate immune responses, and CD8 plus cytotoxic T cells, which destroy infected or cancerous cells, known as cell-mediated immunity. Next is null cells. Null cells are a special group of lymphocytes that lack typical B or T cell markers. They include NK cells, NKT cells, ADCC cells, and LAK cells. Their role is to provide innate cytotoxic defense, meaning they can kill infected or tumor cells without prior sensitization of MHC restriction. Phagocytic cells, the phagocytes such as macrophages, neutrophils, eosinophils, and dendritic cells are crucial for the initial immune defense. They engulf and destroy pathogens through phagocytosis and also present antigens to lymphocytes, linking innate and adaptive immunity. Next is major histocompatibility complex, MHC. The MHC molecule classified into class 1, class 2, and class 3 play a central role in presenting antigens to T cells. Class 1 molecules interact with CD8 plus cytotoxic T cells, class 2 with CD4 plus helper T cells, and class 3 encodes immune mediators like complement proteins. This system ensures proper communication between immune cells during an immune response. Last is MHC polymorphism, explain graft rejection and disease susceptibility. So for question parts, 3-mark question. First, explain what are the primary and secondary lymphoid organs. Give examples. Second, explain the process of thymic involution. Third, state the main function of the spleen. Fourth, what is the function of malt. Fifth, define plasma cells and mention their key features. Sixth, what are the main functions of macrophages. Seven, differentiate between B1 and B2 cells. Eight, what is the role of NK cells in immunity. 9, write a note on MHC class 1 molecules. And 10, define phagocytosis and name the main phagocytic cells. For 5-mark question, first describe the structure and function of the thymus. Second, explain the structure and immune role of lymph nodes. Third, discuss the structure and function of the spleen in detail. Fourth, compare and contrast primary and secondary lymphoid organs. Fifth, explain the process of T cell maturation and selection in the thymus. Sixth, write short notes on different types of T lymphocytes and their function. 7, discuss the mechanism of antigen presentation and MHC restriction. 8, explain the process and stages of macrophage activation and antigen processing. 9, describe the structure, development, and function of B cell. And last 10, write a note on MHC classes and their clinical significance. So with this, we completed whole topic of immune system, in which we studied about B lymphocyte, T lymphocytes, and natural killer cells. So that's all for today's class. Thank you.